Welcome to our lecture on the diversity of chordates, specifically focusing on the classification of cyclostomes. Today, we are going to journey back in time to explore the most ancient living lineage of vertebrates. These fascinating, jawless creatures offer us a direct window into what our earliest ancestors might have looked like millions of years ago, long before jaws even evolved. To begin our exploration, imagine you are swimming in the ocean and you encounter a creature that looks very much like an eel. But when it turns around, you notice something shocking. It has no jaws. Instead, it has a perfectly round, suction cup-like mouth filled with tiny, sharp, horny teeth. This is a cyclostome. The word cyclostome literally translates to round mouth, which perfectly describes their defining feature. Understanding these creatures is crucial because they represent the oldest living lineage of vertebrates. They give us a living, breathing model of early vertebrate evolution, showing us the anatomical baseline before jaws and paired fins entered the evolutionary picture. Now that we know what a cyclostome looks like, let us look at how biologists formally classify them. Cyclostomes belong to the superclass Agnatha, which encompasses all jawless fishes. Within this group, there are two distinct, currently living classes. First, we have class Myxini, commonly known as hagfishes. These are deep-sea marine scavengers, famous for producing massive amounts of slime. Second, we have class Petromyzantida, or the lampreys, which are often parasitic and use a highly developed sucking oral disc to attach to hosts. While both classes share primitive traits, like a cartilaginous skeleton and the complete absence of paired fins and scales, their vastly different lifestyles and anatomies require them to be separated into these two distinct classes. Understanding their classification naturally leads us to ask why these slimy, jawless fishes matter in modern biology. From an evolutionary standpoint, cyclostomes are the crucial missing link. Studying them helps scientists understand how the vertebrate brain, skull, and immune system first developed millions of years ago. Beyond evolution, they have a massive real-world ecological and economic impact. For example, sea lampreys have become a highly destructive invasive species in the Great Lakes of North America. Their parasitic feeding habits have devastated commercial trout populations, costing millions of dollars. On the other hand, hagfishes are incredibly beneficial. They act as the garbage disposals of the deep ocean by consuming dead whales, which prevents disease spread and recycles vital nutrients. To manage these ecological impacts, field biologists must be able to accurately identify these creatures, which brings us to the mathematical tools of taxonomy. Instead of just guessing, scientists use a taxonomic trait vector to systematically classify organisms based on observed anatomical features. The formula shown represents a specific trait vector for an unknown specimen. In this vector, the first variable, G7, represents the presence of exactly 7 pairs of gill slits. The second variable, EF, stands for functional eyes. The third variable, DS, represents a sucking oral disc. By mapping out these variables, a biologist can create a precise mathematical profile of an animal. If any of these physical traits change or are missing, the variables in the vector change, leading to a completely different classification outcome. Let us see this trait vector in action with a concrete example from the field. Imagine a marine biologist discovers a jawless, eel-like vertebrate in a shallow river. We need to determine if it belongs to class Myxini or class Petromyzantida. First, we observe exactly 7 pairs of gill slits, which matches our G7 variable. Next, we note well-developed functional eyes. This is our EF variable, and it immediately rules out hagfishes, which live in the dark and have nearly blind vestigial eyes. Finally, we observe a distinct sucking oral disc, our DS variable, which is the hallmark adaptation of a lamprey. Because our specimen perfectly matches every variable in our trait vector, we can confidently classify this unknown fish into the class Petromyzantida. Now that we have classified a single specimen, how do we mathematically compare two different classes? This is where the Jacquard Similarity Index comes in. This formula provides a strict logical framework to quantify how similar two organisms are, removing human bias. The variable J represents the index itself, which is always a decimal between 0 and 1. The variable C stands for the number of traits shared by both organisms. The variables A and B represent the total number of traits present in the first and second organism, respectively. If the number of shared traits, C, increases, the value of J gets closer to 1, meaning the organisms are highly related. If C is very low, J drops closer to 0, justifying their separation into different groups. With our formula defined, let us apply it to a specific example, comparing our two cyclostome classes. A university student is analyzing a simplified morphological trait matrix to understand the evolutionary divergence between a hagfish and a lamprey. We are given that the hagfish has a total of 8 distinct traits, so A equals 8. The lamprey has a total of 9 distinct traits, meaning B equals 9. Upon close examination, the student finds they share exactly 3 traits: a jawless mouth, a cartilaginous skeleton, and the absence of paired fins. Therefore, C equals 3. Our goal is to calculate the Jacquard Similarity Index to determine their taxonomic overlap and see if the math justifies separating them into different classes. Now that we have our values, let us begin the mathematical proof of their divergence by setting up the calculation. We start by substituting our known values into the Jacquard formula. The numerator is simply our shared traits, which is 3. The critical part of this proof lies in the denominator. First, we add the total number of traits for both organisms, 8 plus 9, which gives us an initial sum of 17. However, if we stop here, we are counting the 3 shared traits twice, once for the hagfish and once for the lamprey. To find the true number of unique traits across both groups, we must subtract those 3 shared traits from 17. This logical adjustment gives us a final denominator of 14. Continuing with our proof, we now have a simplified fraction of 3 divided by 14. When we calculate this, we get a final Jacquard index of approximately 0.214. In physical terms, this means there is only about a 21.4% similarity between the hagfish and the lamprey, based on this specific trait matrix. Because this similarity index is quite low, it mathematically supports the biological consensus that these two organisms are highly divergent. Even though they are both jawless cyclostomes, this mathematical proof demonstrates exactly why taxonomists place them into 2 completely separate classes rather than grouping them together. Their evolutionary paths split hundreds of millions of years ago. To visually reinforce what we have just proven mathematically, let us look at this anatomical diagram comparing the 2 classes. On the left, you can see the hagfish, representing class Myxini. Notice the fleshy mouth tentacles at the front, which it uses to navigate the dark ocean floor, and the slime glands running along its side, which are unique to its scavenging lifestyle. On the right is the lamprey, representing class Petromyzantida. Here, you can clearly see the prominent circular sucking disc filled with horny teeth, designed for latching on to other fishes. Just behind the head, you can count the exactly 7 pairs of gill slits we discussed earlier. This diagram perfectly illustrates the 21% similarity we calculated. They look fundamentally different. We have covered a lot of ground today, so let us summarize our key takeaways. First, we learned that cyclostomes are ancient, jawless fishes belonging to the superclass Agnatha, representing the dawn of vertebrate evolution. Second, we saw that despite shared primitive traits, they are divided into 2 highly distinct living groups, the scavenging hagfishes and the parasitic lampreys. Third, we explored how biologists use mathematical tools, like trait vectors and the Jacquard Similarity Index, to accurately classify these creatures and prove their deep evolutionary divergence. You now have a solid understanding of both the biology and the mathematics behind cyclostome classification. Great job following along with these complex concepts.